Hi, my name is Mike Vaccaro, and today we're going to be talking about a better method for laboratory analysis of glucose metabolism. In order to understand the importance of this new method for understanding glucose metabolism, we first need to look at glucose metabolism. Glucose metabolism can happen in two ways, a catabolic and an anabolic pathway. The catabolic pathway is glycolysis. Glycolysis breaks down glucose to produce 2 ATP, 2 NADH, and either 2-pyruvate or lactic acid. Glycolysis in an anaerobic condition will result in the production of lactic acid, and glycolysis in an aerobic condition will produce 2-pyruvate molecules. These 2-pyruvate molecules will go on to the TCA cycle and eventually produce more NADH ATP as well as FADH2 to be incorporated into the oxidative phosphorylation and result in greater production of ATP. In the absence of oxygen, gluconeogenesis often occurs. Gluconeogenesis uses the byproducts, mainly lactic acid, of glycolysis and produces. On the contrary to glycolysis, gluconeogenesis is the anabolic process which uses byproducts from glycolysis, such as lactic acid, in order to produce glucose. This usually occurs in the liver. An important disease associated with glucose metabolism is diabetes mellitus type 2. The reason why diabetes mellitus type 2 is important is because it's an adult-onset type of diabetes that affects over 30 million Americans today. Characteristic of type 2 diabetes is low insulin sensitivity. Insulin is a hormone that is secreted by the pancreas from beta cells and attaches to receptors on somatic cells, inducing the influx of glucose into the cell. In people with type 2 diabetes, their cells are less responsive to the insulin that is produced. Thus, there is less incorporation of glucose into the cell. Some common causes of this are obesity, lack of exercise, and diet. Often high-fatty foods, as well as high-sugar foods, will result in this insulin tolerance and therefore low susceptibility of cells to insulin. This insensitivity to insulin results in high blood glucose, which can have negative effects on many organs, such as the kidneys, the heart, the eyes, as well as the nerves. Type 2 diabetes is a disease that can be managed through the change in diet, increasing in exercise, as well as pharmacological agents. Although this disease can be managed, there is currently no cure for the disease. Glucose-insulin clamps are methods used to discover an individual's glucose metabolism rate as well as an individual's sensitivity to insulin. In order to do this, researchers use an isotopically labeled glucose molecule and infuse it into an animal model. Currently, there are two different glucose-insulin clamps that are used in order to observe glucose and insulin metabolism and resistance, the first being the hyperglycemic clamp. When doing the hyperglycemic clamp, researchers raise the glucose level to higher than the optimal glucose level based on your body size and weight. They then continuously keep an infusion of glucose into the blood and measure levels of blood sugar content. Over a period of time, they observe how much glucose they have to infuse into your blood in order to keep this blood glucose level at its high level. The data collected from this type of study indicates an individual's glucose metabolism rate. The second type of glucose insulin clamps are called hyperinsulinic euglycemic clamps. During the process of the hyperinsulinic euglycemic clamp, insulin levels are raised to levels that are higher than normal in the individual. Over a period of about 2 hours, glucose is infused into the individual. Blood glucose levels are monitored every 5 minutes, and an appropriate amount of glucose is infused into the individual in order to maintain a steady state. This test therefore analyzes an individual's sensitivity to the hormone insulin. The paper that we're going to be talking about today is titled Quantifying Rates of Glucose Metabolism After Intraperitoneal Bolus by Wang et al. In order to understand the purpose of this study, we first need to look at the method that's currently used today, the intravenous bolus injection. An intravenous bolus injection is an injection through the vein in which a bolus or a sample of a medicine or glucose tracer. This is a tedious and time-consuming method as it requires a catheter insertion. Catheter insertions typically require surgery and a recovery period of about a week. There's a high mortality rate following the surgery or during the surgery and when the catheter is inserted, it requires continuous flushing throughout the trial period in order to ensure that the catheter does not close up. Another method used for injections into rat and mice models is the intraperitoneal injection. This injection inserts a needle into the peritoneum, which is the serous membrane surrounding the abdominal cavity, and it injects whatever fluid that they're studying into the. This method is typically used when an intravenous option is not available. Because an intravenous option is available for monitoring glucose metabolism, this intraperitoneal injection has not been studied for understanding glucose metabolism. One thing to consider when doing an intraperitoneal injection is that the injection needs to enter the central cavity of the organism. This is important in ensuring that the injection diffuses throughout the entire body. The main purpose of the study was to observe if an intraperitoneal injection of a bolus would act in a similar manner to an IV injection of that same bolus. Two boluses used were the isotopically labeled glucose as well as pharmacological agents, which we will talk about in a bit. The final purpose of the study was to see if an intraperitoneal injection of a bolus, along with a mathematical computing model, could replace the use of a glucose insulin clamp in determining an individual's glucose metabolism as well as an individual's insulin sensitivity. The first test that was performed was an IV and IP bolus injection of isotopically labeled glucose into rats and mice. This was done after a 5-hour starvation period on the rats and mice. This starvation period is important because it allows for gluconeogenesis to start occurring, which induces production of glucose within the cells. Administration of isotopically labeled glucose allows us to determine a dilution of the isotopically labeled glucose in terms of the entire glucose pool, therefore allowing us to determine the total glucose production. The second study done was performed on diabetic mice and rats. These mice were fed glitazones, which are drugs that help to increase insulin sensitivity in diabetic patients and mice. Once again, there's a starvation period of about 5 hours, and then an IP bolus injection of isotopically labeled glucose was injected into the mice. Using the same methods previously described, the researchers were able to determine the rate of glucose production in these mice that were fed these glitazone drugs. The first study that we're going to look at is a comparison of intravenous and intraperitoneal bolus injections as a method for introducing glucose into both mice and rats. In Figure A, we're looking at glucose concentration in milligrams per deciliters observed over a period of time. As you can see, regardless of use of IV or IP injection, glucose concentration remains relatively similar throughout the trial period. In Panel B, we're observing glucose enrichment from the isotopic glucose that's added into the cells. An observable decrease over time is seen with the glucose enrichment in the cells. This is because as glucose production increases in the cells, the isotopically labeled glucose concentration decreases relative to the entire pool of glucose. When comparing the data from the IV and the IP injection, you can see that there are similar results observed, thus indicating that the intraperitoneal injection could be useful in laboratory experiments involving glucose enrichment. In summation of the paper, regarding other figures as well as what was previously shown, the researchers concluded that an IP bolus of glucose tracer is indistinguishable from an IV bolus in rat and mouse models. Through the use of glitazones, the researchers also observed that an IP bolus injection is sensitive to currently known diabetic treatment through pharmacological agents. The researchers also showed that an intraperitoneal bolus injection can serve as a proxy for the tedious glucose insulin clamp method. One must keep in mind that an intraperitoneal bolus injection also requires the use of a mathematical model in order to calculate the total amount of glucose within the pool. The results from the study are important because they provide information regarding a new method that is more efficient for understanding glucose metabolism in rats and mice models. Data from these sorts of studies is important to each individual suffering from diabetes, so increasing the efficiency of these laboratory methods is important. The main purpose of the study was to observe if an intraperitoneal injection of a bolus would act in a similar manner to an IV injection of that same bolus. Two boluses used were the isotopically labeled glucose as well as pharmacological agents, which we will talk about in a bit. The final purpose of the study was to see if an intraperitoneal injection of a bolus, along with a mathematical computing model, could replace the use of a glucose insulin clamp in determining an individual's glucose metabolism as well as an individual's insulin sensitivity.